they're pretty straightforward. And then this one is a little bit more complicated to make. Then we have an animation based on a static structure, animation based on a trajectory, and then lastly we use some more sophisticated techniques to go through to sort of fade smoothly between different representations. Okay. And if you guys catch anything wrong with any of the steps of that tutorial, please tell me today. That would be really helpful. All right. Okay, so you can, I don't know so I'm guessing, different people here have different levels of expertise with VMD, so I'll probably err on the side of over-explaining things. If there's a specific step that you guys don't understand or it's not clear why I'm doing it, then please ask a question. Okay, so by default, only these two windows will come up in the beginning. One that has the display, and one that we call the main window. Also, you guys can try to follow along if you want. Now, there's two other windows we recommend you open right away, which is representations. And also the TK console. Now with the TK console, you can run an individual command, and you can also source a script. I actually won't be sourcing any script today, but be aware of that functionality. All right. So when you start out, the default will be a perspective view, so things that are closer to you will appear quite a bit larger. So actually, you can switch that into an orthographic view, so everything will appear the same. You can do rotations and zooming. Right. Now there's one thing that's nice about VMD is you have many different representations. So I'm going to switch from lines to van der Waals. So those are beads that should get the approximate size. And increase that so you can actually see it. So one thing that may not be directly in the tutorials that you can press. So when you're clicked on the display window, you can type C for centering, and then click on a specific atom or bead, and then you'll zoom in on that specific one. Now, and then the rotations will be about that point now, not about the middle. And another thing that you can do is you can change the resolution of a different representation. So I'll go down to show what that looks like, and going back up, you can see that the representation looks nicer then. And there are different materials available. Switch that to AO chalky. So the AO is for ambient occlusion. So there's so actually right now we're in what's called the rotation mode. But you can also switch to translational mode, where if you left click, it'll move it around without rotating. Switch back to rotation mode. And you can also do, in translation mode, you can click on the right button. You can go into and out of the structure like this. So another useful functionality is you can press equals and it'll reset to the beginning at any point. Okay. Okay. So I'll do reset. So From the TKCon, you can also do rotations, and that could be nice if you always want to do like a 90-degree rotation. So you do rotate X by 90. So that's a way to do rotations without having to use the mouse. Yes.
Can you mark the TK console so that we can see what you have?
Well, sorry, what's the question?
Can you enlarge the TK console so that we can see what you are typing?
Sure.
Thank you.
So I don't actually no. So unfortunately, so I could increase it, but then the font is still not increased. So sorry about that. So these kind of instructions will be in the document of the tutorial. So sorry about that. All right, and then if there's okay, yeah, I guess I'll have to. If it's difficult to follow along in the PDF, then I can help you out with that. All right. So one nice thing so this is specific to the output of mRDNA, is if you color by the occupancy, that will be so now it'll be colored by the helix ID that was set. And if you color by the beta, then that actually is the nucleotide ID that's set by Cadnano. All right. Now, right now we've only done one selection. So now we'll do two different ones. And you can also you can choose your own color. Now, if you do create grep, it'll copy the same one. I'll do a different one that's not that, and choose a different color. So the way that I have it colored now is the blue beads will be double-stranded DNA, and the yellow will be single-stranded. Now, one thing that you can do is you can actually change the definition of the color. And we often do that with blue. Okay. So anytime I have the TK and then the two forward arrows, this is something that can be copied and pasted directly into the TKCon. So I'll do that. So you can see this changed the color of the blue beads, and actually it also changed the color of this axis. Right. Okay, so a black background can be nice for visually looking something for analysis, but if you want to make an actual image that will go into paper, it's nice to have a white background. So you go to Graphics, Colors, Display, Background. You can choose any color you want, of course, but white is nice, since paper's white. Okay, you can also, so the axis can be nice for doing rotations and things like that. But when you actually make the image, often we'll turn the axis off. So you can do it from the GUI, but you can also, from the TKCon, you can load the trajectory. Mol, add file, and the name of the trajectory. Okay. And you can, so in this way you can watch for yourself the output of the simulation. Now, one thing that you might notice when you first watch this is it seems kind of jumpy or jiggling around. And okay, I'll wait for this to finish. So this is actually a common question that I get from people who have actually used VMD for some time is sort of how can you smooth that? And so there's another tab in your representations window, that's trajectory. And it's this right here, there's smoothing windows. So I'll increase that. And you have to do this for all the different selections that you have, so I'll do 3. So what this does is the first frame that's shown will actually be an average of the first 4. And then the second frame that's shown will be an average of 2, 3, 4, 5. So you can rewatch the trajectory. You can also change the speed at which it's shown. So you can see now that the motion is a lot smoother when you watch this. Okay. You can pause the playing when you're running a trajectory. You can also, so there's these 2 buttons here that will take you to the beginning and to the end. Okay, so now what I'm going to do is make a new selection. Another selection that is everything. I'll do the same smoothing. Now, I want to do a quick surf representation, and depending on the amount of memory that's available on your computer, sometimes that will crash. So there's a button down here that says apply changes automatically. So you want to, if you're worried about a selection break or a new representation breaking something, you can click on that. So now I can do a change, and it won't be done immediately. So I'll switch to quick surf. I'll switch the color to white. I'm going to increase the radius and also increase the grid spacing. So now it won't be as fine. Now I'll do apply. It'll take a little bit of time, but now you can see that there's a surface representation that's on top of the other one. And one nice functionality is that you can, under graphics, you can make a new material for yourself. Now, if you can click on whichever sort of base material that you want to do. If you create new, it'll copy all these presets here. And all I'm going to change is I'm going to decrease the opacity. And then also I'm going to make it so that it has angle modulated transparency. You can change the name of this material also if you want to, but the default is fine. Now I'll switch the material of that third one to the semi-transparent one, and then also you have to apply. So now actually, although it's perhaps not immediately available, or not immediately obvious, there's a representation that's on top of what's already shown. And what I'll show is, first I'll do rendering with the default, which is snapshot. So just file, render. Slider snapshot. So you'll see this will be very fast, but it doesn't really look very nice. Now if I zoom in, but you can actually see that there's some of these like white dots here that, so you really can't see the surface very well. One way to change that is under your display, you can go to render mode, DLSL, and actually already you can tell in this representation that it looks a little bit nicer. If I make another rendering, you can see. Now, what we recommend that you do instead of these two is to use that tachyon renderer. And so first you go under display settings, turn on the shadows, turn on ambient occlusion. And so there's these two, AO ambient and AO direct, which sort of tune the degrees of the ambient light and the direct light. Now, one trick that might be nice for you guys is you can actually, anything that you can do in the GUI, you can also do from the TK-con, and you can log any kind of command that you do into that window. So now I know the exact command that was done when I pressed that button to increase this slider. Now, the only way to increase the AO ambient to a value above 1 is to do it from a command. So I'll do 1.4. Okay. Decrease the direction. And you can turn off the logging at any point in time. And now you guys have access to a Tachyon optics render, but that won't be available on your computer, laptop, unless you have a nice GPU. So I'll do the Tachyon internal. And I'll save that file. So actually, so this rendering will take a little while to do. So it's working on making that image. So this will take about a minute. So one thing I also want to tell you guys about is, so to make this image, what I did was I used the optics renderer that has an interactive display. So one thing that's nice about that is you click on the render button and then you can rotate the image, you can translate and do all those kinds of things, and then it'll interactively show you what the rendering would look like, and then so then you can change things after you already pressed that render button. So this is still going, and so another thing that I would say while this is rendering is that when you're making a movie. When you first make the movie, sort of as practice, you can do a snapshot render. But then when you want to make something that you would put in a paper or presentation, then you can switch to Tachyon. And if you have access to the optic render, then it'll be quite a bit faster, something on the order of like 10 times faster. So this is almost finished. Okay, so you can see, so now I'll just I'll close this and compare the 3 renderings side by side. So, and by default, it will save that that rendering wherever you're located in terms of in that directory. So, show side by side. This is the standard view on the left. This is switching. And then this is with the Tachyon. Now, if you want to modify this third one even further, you can put it into another software and change things like the exposure and the saturation. But I won't go over that today. Now let's do take the picture. Go back to all right. So now I'm going to briefly go over. There's something where you can save different viewpoints and move between them. So you go to Extensions, Visualization, View Change Render, and this will so you can do anything that you want in this window in the display, and you can save that. Okay. Then I'll change it. Save that viewpoint. Zoom in. now, one nice thing is you can click on one of the specific viewpoints and you can go select viewpoint. You can also change the amount of time, so that was quite fast. Now it'll be done more slowly. All right, and so going through these kinds of steps to make an actual movie will be gone over in the tutorial. And so you can add all these viewpoints to the movie, enable the movie maker, change the total time. And then from here you can actually preview the whole thing. And then, okay. Now to actually make the movie, so let's finish. To actually make the movie, there's another extension called Movie Maker. So I'll go to visualization again. Movie Maker. So you get to choose the renderer. By default, it'll be that snapshot that I showed you first. And then, of course, you can do a trajectory if you want. But in this particular case, we want to do the user-defined procedure. And then you can change the location of where you want that to be. So right now, I'm just working at the demo. You can also change the name of the movie. Okay. So, and then it'll tell you the amount of time that the total movie would be, and then you can just press this. So that's all that I prepared for you guys. Do you guys have any questions? You can ask me now on a specific step, or you can ask me when you guys are working through the tutorial. Are there any questions? Okay. So if there are no questions, then I'll hand it over to Chris. And he made a movie that goes over some things that you can do in VMD with in terms of analysis.
Well, so I just highlighted two really basic, analysis steps that you might want to do pretty much every time you run a simulation. The first is looking at the root mean square deviation, or RMSD, of a structure. I won't hit play yet. The second is looking at the root mean square fluctuations. So RMSD is sort of like a measure of how far the entire structure has changed, how much it has changed from its initial configuration. And that can be useful to tell not always if you've reached a steady state, but at least if you haven't. And RMSF can highlight what parts of the structure are really flexible or fluctuating. And that can be useful if you want to try to optimize the structure. So here we've loaded in a simulation, not a huge one, but a simulation of this polyhedral mesh bunny structure from the Hochberg group. And I'll attempt to narrate what's going on. So the first approach will be to take this bunny and use a plug-in in VMD to measure the RMSD. So you go to the extensions menu, go to analysis, and then there are several RMSD tools. My personal favorite is this RMSD trajectory tool. So you click on that, and you get this slightly overwhelming box. There are a few main things that you need to worry about. The first is that you need to set a selection. Protein is not going to do it for us, so let's just select everything. We've got the reference molecule. It's the top molecule, the only one we have loaded. And usually when you do RMSD, you want to align the entire structure before you actually calculate the RMSD. So we have an align button, RMSD button, but before we click the RMSD button, we want to check this plot box, or you can also check this save box next to it. And then this will give you just pretty immediately a quick view of the root mean square deviation for your structure. And that's great. So the next thing that I'll actually, oh yeah, you can export this to say XF trace, but I won't do that. And you can of course save just the plain data, so you can use whatever plugin software you like. So then the next thing that I'll show, yeah, I guess I'm hovering over average, min, max values. Sorry. The next thing I'll show is how you can do this also from the TK console, because often you do the same analysis over and over. It's nice to have just a little script for it. So the first thing is to go to the extensions menu and pull up the TK console. And then from there, you can create an atom selection that you will measure the RMSD against. So I'm calling it ref here. So we've set a variable with an atom selection containing everything at frame 0. And then we'll create an atom selection that will loop through, and that's that I've called SEL, or cell. So the current frame for cell is now, which currently we're at the end of the trajectory. So it'll track wherever you are in VMD. But you can also specify a particular frame. Now, VMD has this measure RMSD command, and you give it 2 selections, and if they have the same number of atoms, it'll tell you sort of how far they are apart in terms of the Rubin square deviation. So you can change the frame, and so you get a different value for RMSD. Then the next step is to open a file that will print statements to about the RMSD. And then finally, we will run a loop over the frames in the system, updating the selections frame every time, and then printing a line to the file. So this mulinfo top get num frames is how you say, okay, there's 101 frames. That's how you can do it programmatically, so that this script would work regardless of how long your trajectory is. Yeah, so that's what a loop over all the frames looks like. Each time we enter this loop, we'll update the frame for the selection, and then we'll print out two values in each row for each line of the file, the frame and then the RMSD at that time. And then finally, you want to close the file, so that'll flush all the buffers. So then you can take that file and plot something. So finally, I'll show the RMSF. So it's very easy to obtain the RMSF, but it's quite overwhelming to look at. You get a number for every single particle in the system or in your selection. So instead of just printing it in a file, we will color our structure by setting the beta field to the RMSF. So that's how you do it. And then from there, you can go over to the graphics menu, which you get to here, if you don't already have it up. And then we'll go to the coloring method, and we'll change it from the name of the atom down to beta. And then we get this map that looks like this. Red is low and blue is high by default. And you can change the range in the trajectory tab. So right now we're going from 7 to 35, but it's nice to start at 0. So red is 0. And then we can take a peek at our structure and see what parts are deviating most strongly or fluctuating most strongly. Maybe those are problematic parts of the structure. Yeah, and then also you can if you don't like this color scheme, you can go to the graphics color tab and pick a different scheme. And actually, it's also possible to define your own colors for the map. But I think that has to be done from the TK console. Maybe not. Maybe there's a way. Maybe I'm not sure. So this is the part of the structure that's fluctuating the most, and we can see that here, and we can play the trajectory. We know that that's the part of the structure that we want to look at, and that's basically it. This little movie. Yeah. So if you have more complicated analysis that you need to do, I'm happy to talk to anybody about what it is you're trying to achieve. And VMD is a great tool for these things. It's not the only tool out there, and I generally recommend using whatever's simplest for you to use. But I'm a fan of VMD because you get the visual thing, and you also can code things, script. PCL is not my favorite language, but it's it's pretty complete. So you can still do anything you need. Yeah, so that's it. Thanks. Yeah, so let me run my laptop. Switch. Let's not do that again.
Yeah.
No, I'm going to try to do this. Yeah, we're running a little behind, but hopefully that's okay with everyone. We'll get you to your coffee break soon. All right, so for the last talk of the session, I'll tell you a little bit about how we can use our software tools to integrate with other software. The first thing that we've kind of already gone over a little bit, you have on your Amazon image, and you can also download this freely. You have a Python package called mrdna, or multi-resolution DNA, that includes a couple of scripts that can be used to run coarse-grained simulations or atomistic simulations with our center softwares ARBD and VMD, respectively. And the mrdna script can, we've already demonstrated yesterday, it takes in cadmium as input. But we've recently also extended this to take in eHelix Maya files, which is how we had that simulation of the bunny I just showed you. And finally, we've recently, very recently, added a PDB reader, which opens up a lot of possibilities for integrating with other kinds of software. And so the usage of different inputs is covered in a tutorial. It's our universal all-atom tutorial. And it specifically covers how you use the mrdna script to run actually NAND simulations, or to prepare NAND simulations. Yeah, so you've kind of already seen this, but I'll show it anyway. So this is our one of our favorite polyhedral mesh structures. This bunny. It's not spring anymore, but I saw one last night on my way home, so they're still around. They're still relevant. Yeah. Okay. So, yeah. So so besides cadnano and eHelix, we also have this PDB reader, and in principle, you can use this to take output from Candu, Daedalus, Oxdna, Nucleic Acid Builder, TMA, and Nano Engineer 1, which all either have native PDB output or conversion utilities. But depending on it's a new tool, depending on the size of the structure and how canonical the DNA in it is, it may or may not work. Give me feedback. I'd like to know what's working, what's not. And for some of these, we definitely plan to add a more native input, for example, Daedalus. Okay, and again, this is covered in our universal all-atom tutorial, which is available on GitLab. It's also available on the center's workshop page under the tutorials tab for this workshop. And it's available on your Amazon image from the TCBG tutorials folder desktop. So feel free to give that a try. And all of these inputs, they don't just have to be used for creating a simulation with NAND. You can also create one of these multi-resolutions with any of those inputs. Okay, so now I'll talk about how the simulation tools that we have can be interfaced with other kinds of software. So first I'll talk about the coarse-grained simulations. So I kind of covered this yesterday actually, but our coarse-grained simulation software can interface with potentials that are obtained from continuum software packages such as Console, which is what this is showing here. So this is a heat map of the potential inside of and around a nanopipette, which is incorporating the mobility of the ions, the electronegativity of the glass of the pipette, and these physics. I think they call it multi-physics. So we can take this sort of 2D result and create a three-dimensional potential. And there's a very particular format for this that we take, but it's a simple one to create actually. It's simple but particular. And then we're able to run these simulations of capture. And if you do it at too high of a bias, your structure will get pulled through this plate. And then we're able to look at, with a higher resolution model, at thread efficiencies at different biases, with the force coming from essentially a continuum model. But it's not dynamically coupled, so that's one caveat. And from this, these simulations, we're able to get distances between the labeling sites, and from those, we're able to estimate thread efficiencies that agree pretty nicely with the experiments. So we're really quite happy with that. And this is all possible because the software package that we are doing this simulation with is from the ground up designed to take in three-dimensional tabulated potentials. So potentials that you can look up, basically. And that's been used, so it's a new piece of software, but it's been used already a bit for various projects in the center, including a study of cytochrome C2 diffusing through the electrostatic field inside of a chromatophore, and in a study of a small patch of cytosol. It was used actually to set up initial conditions for an all-atom simulation. We've got some preliminary modeling of chromatin, but that's really, I would say it's not a, we've got a lot of work to do there actually. And then David has almost completed a project studying the nuclear pore complex NG nukes, which are inside of a rigid. So the disordered parts of the protein are modeled as beads, and they're inside of a field produced by this rigid scaffold around the or that forms the actual pore. And I've also used this tool to study a biological process in bacteria, which is replication actually. This image is from good cell, of course. And if you look at the replication fork, there's this little protein called single-strand DNA binding protein, and I wanted to know sort of what are its dynamics. We have experimental collaborators looking at it in single molecule experiments. We wanted to see if we could reproduce those. So we ran some coarse-grained simulations with a DNA model interacting with a rigid body model of the protein. And a lot went into this, into parameterizing this, so you can't just pick up any protein and apply this kind of modeling. We had to do a fair amount of sort of optimization of a coarse-grained map of the protein that was based on an all-atom simulation. So it's trying to reproduce data from all-atom simulation. But it worked really pretty well. We got acceptable agreement with our experimental collaborators, so we were kind of happy with this. So this is a force extension curve. It's actually the difference of the force extension curves between constructs with and without this binding protein. So that's just kind of showing a little bit of capability of our coarse-grained package. But actually, MD also has a very similar mechanism for applying potentials. And we've used that in 2 projects that I'll highlight now. But before I get into those projects, I guess I want to remind you that one of our one of our main areas of research has been looking at DNA passing through nanopores in the hopes of sequencing the DNA by modulations of the ionic current. So this is a problem that Alex has been looking at for a long time. And several years ago, a few papers came out that showed that plasmonic nanoparticles use to locally heat nanopores and modulate the ionic currents. And actually, when you heat these pores, you get an increase, like a doubling of the current. So that's kind of an interesting thing. This was done with both biological nanopores and solid state. And we thought, well, what are the effects of local heating on DNA in a pore? And so we ran some continuum simulations of a nanopore that has sort of an annulus of a conducting material that would heat up when you shine a laser on it. And we looked at the dissipation, how that heat dissipates in space. And so we ended up getting these temperature maps, and we just ran all atom simulations of DNA in a nanopore with the temperature gradient like this applied. And what we found is actually that the DNA gets stretched out, which is maybe a little bit counterintuitive actually, because normally the classic picture of a polymer is that heat causes them to shrink. But what we have is not just heat, but a high temperature. We have a temperature gradient, and there is an effect called thermophoresis that just emerges when we all have simulations, whereby small objects move in a temperature gradient. And this isn't my project, so I actually don't know all of the background behind thermophoresis. So I won't be able to answer complicated questions about it. But what we saw in our all-atom simulations is that there was a piconewton level force on the DNA. These were pretty high gradients. I think, these were pretty high temperature gradients actually. But we thought that was interesting, so we went to a coarse-grained model again, and we took the force that we had established was applied to each nucleotide in a temperature gradient, and we looked at systems where one end of the DNA molecule was confined in a pore, and we had 2 simulations. On the left, there's no heating, and on the right, we have this thermophoretic force, and you can see that the DNA becomes more stretched in the latter case. And that was kind of exciting to us because you want your DNA to be stretched in order to sequence it. That's roughly the idea. And it also, sequencing is aided by the increased current that you get. So this would be, a nice thing to have for solving applications. And I'm not sure what the experimental challenges of creating, an annulus around a nanopore, a conducting annulus around a nanopore are. So that might be quite a challenge. Now, around the same time, we ended up collaborating with Case Decker, who has these who created these bow tie plasmonic nanostructures surrounding the pore. Again, we had an eye towards sequencing applications. So when you shine laser light on these gold nanoparticles, you get 2 things. You get a current through, but you also might get trapping of the DNA at the gold surface. And finally, and the reason for that is that the plasmonic field has a really sharp electromagnetic intensity gradient, and that exerts a force on a dielectric material. So you might get tracking of the DNA, and you also might, because of the high intensity of the field, you might get what's called surface effect Raman scattering. I might have that wrong. So you might actually be able to get an optical signal that tells you something about what's in the high intensity region of the field, of the plasmonic field. So we wanted to run some sort of proof of concept simulations of this. And so we simulated this gold nanostructure in the lower right here using an FT, finite domain time difference method that we then, so this gives you basically the electric field as a function of time. And we took the average intensity of that and as an output and fed it into a model for how it should apply forces to the DNA that could be applied in our all-atom simulations using, again, these sort of tabulated grid potentials, these three-dimensional potentials. So what you find is that you get trapping forces in very close proximity to the gold nanostructures. And in simulation, we visualize that as follows. These arrows are showing the trapping force. And we were able, in simulations, to successfully trap these DNA structures. And we had trapping in a single location. We also had situations where the DNA would sort of buckle and curl and get trapped at each bow tie structure, at each of the triangles. And we found that the displacement of the DNA slowed with the intensity of the infinite laser light. So that was gratifying. And so then the final thing that we looked at is whether or not it would be, in principle feasible to trap, release, and then trap, and then from the trapping to generate a signal that you could potentially see. And I don't know the details actually of what's behind these plots, so I apologize for that. Again, this wasn't my work. But I think that the gist was that, in principle, yes, you should be able to see a SIRS signal from the DNA that would allow you to distinguish between individual nucleotides. But in practice, I think there are challenges. So the last project that I want to tell you about also came out of a collaboration with Case Decker. And this was also using our coarse-grained simulation package. And here we created a sort of idealized double pore system with a sort of impenetrable barrier. And this was again done with these tabulated potentials. So you can say, okay, I want this region, this bilayer, to have a high potential so that nothing can enter it. So you can also kind of generate idealized potentials from your mind and apply them in simulations. This work was done by Shuhan, who was a graduate student here. So we have two examples. In the top, we have just a DNA strand passing, a single DNA strand passing through a single pore. And then in the lower simulation, we have a double trapping event. And what we found is that this was actually quite long-lived because there's sort of a tug of war going on. There's force pulling down on the DNA from the applied bias. I forgot to mention that's also in this potential. So there's an electric force pulling down on each side of the DNA, and it takes actually quite a long time for it to fall out. And that's a good thing for any kind of sequencing applications. So we were excited about it. So Shuhan did this 2,000 times, and here he's showing sort of the probability density as a function of time. So what you have is that is a situation where most of the strands just pass right through the core, and that's shown here in a distribution. So you get kind of a nice Gaussian distribution for the translocation times, although I think it's on a logarithmic scale, so I guess it's not Gaussian. But if you look several standard deviations away, you have quite a few events that are really long-lived, and that's sort of interest. You know, those are what you would want for sequencing applications. And so when we showed this to Case Decker, they were able to actually replicate this experimentally, and I won't discuss that aspect of it. And they also were able to see this sort of like tail of long dwelling trapping events. So I think you know, overall there is some nice agreement over the details for distributions are really good. Yeah, and they looked at different distances of nanopores. I think that's okay. So I'd like to basically thank the center and my group members for all their great work. Thanks for listening. And if you have any questions about how you can apply potentials in your to your DNA structures, I'd be happy to talk about that on an individual basis. And then also the sort of basic thing, the basic application of an external potential is covered in this multi-resolution DNA nanotechnology tutorial. So you can look at the very end of that tutorial, step 5, and there's an example of applying an external potential. So have a look at that, and that's it. So I guess at this time, I think we're scheduled to have a coffee break.
Yep.
So I don't know if Joel's probably made coffee and maybe some snacks so we can come down to the 3rd floor, take a break, and then come back. We'll have a workshop for the rest of the day. Hands-on tutorial sessions for the rest of the day. And thanks. There's